The topic for the present presentation is cells of the nervous system. A student is expected to enumerate the cells of the nervous system, should know the features of the various types of neurons and the glial cells, should know the morphology of the synapses, the neurotransmitters, and the blood-brain barrier. The cerebral cortex contains neuron and supporting cells. The supporting cells are the glia, ependyma, and the endothelium of the blood vessels. The glia are of three types: the astrocyte, oligodendrocyte, and the microglia. The ependyma are ciliated or the cuboidal epithelial cells lining the ventricles and the central canal of the spinal cord. They form the brain CSF barrier. Tanycytes are special type of cells in the third ventricle that carries hypothalamic releasing factors to the posterior lobe of the pituitary gland. Billions of neurons are there in the nervous system. They are mostly multipolar, each pole giving rise to a process, a dendrite. One of the poles gives rise to origin of the axon. The soma contains the nucleus and cytoplasm with various organelles. Clumps of rough endoplasmic reticulum are known as the Nissl bodies or Nissl substances. Nissl substances are pores in the motor neurons and fibers in the sensory neurons. All parts of the neurons are permeated with microtubules and neurofilaments. The other organelles are the mitochondria, ACR, and lots of ribosomes. The initial part of the axon is non-myelinated and it needs special mention because here the synapses are formed and this is the place for formation of the axon potential. Various types of neurons are present in the nervous system. The neurons may be unipolar. For example, the amacrine cells of the retina where there is no axon. Pseudo-unipolar, where one process is there which divides into central and peripheral part. The pseudo-unipolar neurons are present in the dorsal root ganglia and the trigeminal ganglia. The bipolar neurons are present in the retina. The rest of the neurons are multipolar neurons, and in between the large multipolar neurons lies small multipolar neurons which are known as interneurons. The Purkinje neuron needs special mention because they have very big dendritic tree. The motor neurons of the spinal cord contain coarse Nissl granules. Dendrites are short branch processes which may be multiple in number. The dendrites divides and subdivides to form the primary and secondary and tertiary dendrites. The smaller branches bear number of minute projections which are called dendritic spines which participate in the synapse formation. The dendritic surface also participate in the synapse formation. The axons are single long process arising from the base of a neuron. The length may be up to 1 meter. The axon contains all cell organelles except the nasal granule. It gives off collateral branches perpendicular to the main axon. The axon terminates into small knobs or synaptic buttons, telodendria, which form synapses with a muscle or another neuron. Neuronal proteins are synthesized in the nasal body and packed in the Golgi apparatus. The vesicles from the Golgi apparatus, mitochondria, the neurotubules are transported to the axonal terminal or the terminal button through exoplasmic transport. There are anterograde transport and retrograde transport. There are fast and slow transport mechanism. The fast transport is about 1 meter per day, whereas the slow transport is 5 to 10 millimeter per day. And this transport occurs with the help of microtubules and a protein dynein. The worn out material reuptaken from the synapses and the neurotransmitters use the retrograde transport. The poliovirus and the herpes virus reach the cell body through retrograde transport. The axons may be covered by myelin sheath. The process of formation of the myelin sheath is known as myelination. Myelination is done by oligodendrocytes in the central nervous system and the Schwann cells in the peripheral nervous system. The outer and inner membrane of the oligodendroglia or the Schwann cell form an area called mesoaxon. The mesoaxon spirals around the nerve fibers or the axons to form the major and minor dense lines seen in the electron microscope. The non-myelinated neurons are covered by single layer of axonema sheath from the oligodendrocyte or the Schwann cells. A segment of the axon is not covered with a myelin sheath and which is important for nerve action potential is the node of Ranvier. Any injury to the axons give rise to changes in the proximal segment and the cell body of the neuron. There is swelling of the cell body. The nucleus is placed eccentrically and the nasal granule become finer. The process is called chromatolysis. The endoneural sheath formed by the Schwann cells are essential for regeneration of the peripheral axons. Limited growth or regeneration of the axons of the central nervous system occur as oligodendrocytes do not form a neuronal tube. Two or more neurons and their processes come in contact to form the synapses. Synapses are of two types. chemical synapse and the electrical synapse. The electrical synapses are gap junctions between the neurons or their processes. No neurotransmitter is involved in the electrical synapses. That is why they are very fast. In human brain, these synapses are very rare. It is present in the inspiratory center where synchronization of the actions are required. Morphological types of synapses are axodendritic, axoaxonal, axosomatic, dendrodendritic, and several other types. Classically, the axon terminal ends as a synaptic buton, which contain synaptic vesicles, mitochondria, and neurotubules. The contact site has thick presynaptic membrane, the postsynaptic membrane, and a synaptic cleft. The postsynaptic membrane may be part of the axolemma or may be part of the cell membrane of the neuron. The synaptic vesicles are synthesized or formed in the cell body from the Golgi apparatus. They are transported to the axonal terminal by axoplasmic transport with the help of neurotubules, and they fuse with the presynaptic membrane and release the neurotransmitter. The transmitter molecule cross the synaptic cleft and activate the receptors in the postsynaptic membrane. This event depolarized or hyperpolarized the postsynaptic neuron, resulting in excitation or inhibition. Majority of the synapses use acetylcholine, glutamine, and serotonin as excitatory neurotransmitters. GABA and glycine are used as inhibitory neurotransmitters. There are other several catecholamine and peptides which are used as neurotransmitters and neuromodulators. The types of neurons present in the cerebral cortex are the giant pyramidal neurons or the base cells, the granule cells, the stellate cells, the fusiform cells, the cells of Martinotti, and the Cajal-Redgier cells. The dendrites of the pyramidal neurons and the other neurons project towards the superficial layer, whereas the axons go downwards and form the white fibers of the cerebral cortex. The cell layers of the cerebral cortex from superficial to deep are layer 1 or the molecular layer, which contains the terminal branches of the dendrites and axons of the deeper cells and the Cajal-Redgier cells. The layer 2 are the external granular layer and small pyramidal neurons and interneurons. Layer 3 or the external pyramidal layer, it contains small pyramidal cells, whereas layer 4 is the internal granular layer, which contains the stellate cells, interneurons, and the pyramidal neurons. Layer 5 or the internal pyramidal layer, they contain the giant pyramidal neurons or the base cells and the interneurons. Layer 6 or the multiple cell layer contains fusiform cells, pyramidal cells, and interneurons. The cerebellum contains three layers of neurons: the molecular layer, which contains the dendrites of the Purkinje neurons, and the basket and stellate cells. The Purkinje cell layer, these are the main cells of the cerebellum with a big dendritic tree. The third layer is the granule cell layer, which contain the Golgi cells and few granule cells. The neurons of the dorsal root ganglia and the trigeminal ganglia are pseudo-unipolar, whereas the sympathetic and the parasympathetic neurons are multipolar. The glia has been classified as macroglia and the microglia. The macroglial cells are the astrocytes and the oligodendrocytes. Astrocytes are of two types: protoplasmic astrocyte and the fibrous astrocytes. Protoplasmic astrocytes are present in the grey matter. The fibrous astrocytes are present in the white matter. The oligodendrocytes lie in between the axons or the nerve fascicles. The microglia are very small and they normally lie along with the blood vessels. The ependyma are also classified along with the glial cells, and they form the lining of the ventricular cavity. The astrocytes absorb neurotransmitter around the synapses. They form the internal and external glial limiting membrane. And in the aging brain, the astrocytes contain corpora amylacea, which is a glycoprotein in nature. The food processes of the astrocytes covers the external surface of the capillaries, supporting the blood-brain barrier. The resting form of microglia, which is present in the normal brain, forms 5% of the glial population. They are equivalent to the resident macrophages. They are phagocytic in nature after injury or diseases. Activated microglia retracts their processes. They express antigen and they are mainly seen in AIDS and neurodegenerative conditions. Oligodendrocytes. One oligodendrocyte myelinate 16 axons. They form the myelin sheath in the central nervous system. Satellite oligodendrocytes are small oligodendrocytes in vicinity of the larger neurons. The other cells of the central nervous system are the ependymocytes, tanycytes, and the Schwann cells. The ependymocytes are the simple ciliated cuboidal or columnar epithelial cells lining the ventricle or the central canal of the spinal cord, and they lie in contact with the CSF. They form the CSF brain barrier. The tanycytes are special types of cells which is present in the third ventricle. It carries hormones from the hypothalamus to the pituitary gland. The Schwann cells, they myelinate the peripheral nerve axons. The blood-brain barrier. Vital dyes like India ink, when injected into the ventricular system, stain the brain tissue. The same dye, when injected into the blood vessels, do not stain the brain. The differential properties of the capillary endothelium make blood-brain barrier, which prevents diffusion of the substances from the blood to the brain tissue, and this is known as the blood-brain barrier. The cerebral capillaries are different from the general capillaries. The general capillaries are fenestrated, whereas the cerebral capillaries are continuous capillaries. The capillary endothelium form tight junctional complex which do not allow the substances to pass from the blood to the brain tissue. The anatomical components of the blood-brain barrier are the endothelium of the capillary, the basement membrane, and the food processes of the astrocytes covering the capillaries. The blood-brain barrier is absent in the circumventricular organ, which are the area postima, median eminence, the pineal gland, the subpharyngeal organ, the vascular organ, and the neurophysis or the posterior lobe of the pituitary gland. The area postima is the vomiting center. Toxic chemicals from the bloodstream directly stimulate the chemoreceptor trigger zone, which initiates the process of vomiting. Like all other cells, the neuron contains the nucleus and cytoplasm. The special characteristics of the cytoplasm are the nissl substances, which are the rough endoplasmic reticulum for synthesis of protein. The Golgi complex of the neuron form vesicles. The neurofilaments are also synthesized by the vessel bodies. They form the skeletal support. The neurotubules helps in the anterograde and retrograde transport of the vesicles and the neurotransmitters, and in disease conditions the virus or the bacteria. Synapses are contact sites of neuronal processes with pre and postsynaptic membranes, synaptic clefts and subsynaptic web. There are chemical synapses which are morphologically exodendritic, exosomatic, exoaxonic, and dendrodendritic. The excitatory synapses use acetylcholine, glutamic acid, or glycine as neurotransmitters, whereas the inhibitory synapses use GABA as the neurotransmitter. They are called GABAergic synapses. The electrical synapses are very fast as the neurotransmitters are not involved in the process of transmission. Among the neuroglial cells, astrocytes are supportive, nutritive, and form blood-brain barrier. They are the main source of brain tumors. The oligodendrocytes form CNS myelin sheaths, which are subject to destruction in demyelinating diseases. The microglia are the potential phagocytes.